So I'll stay right here.
Yeah, you can walk maybe like halfway across the screen and it'll still pick up just fine.
Okay.
Just don't go all the way over on the other side.
Just talk really fast.
Yeah. Absolutely.
So I will change the lighting after the presentation.
Okay.
Until he introduces you.
You have a thing of water?
Yes, I
Oh,
can.
do you have water?
Thank you.
She's been at IALR now. Has it been up to 4 years?
It's, well, I did my PhD work there. So
That's true. Yeah, but I mean as director is what I mean.
Yeah, 2 years as director.
It's 2 years.
Mhm, Chief scientist for a couple of years now.
Oh, I see.
Yeah.
So then you were chief scientist when, what was his name? What was the guy before you?
Barry Flynn?
Barry Flynn.
No, I wasn't.
You
I
weren't.
was. No, I was a scientist.
Okay. But you were there when he was there?
Well, my PhD work. Yes.
Yeah.
Mhm.
Should be all good.
Okay.
We switched our projector back to the normal one, so let me know if there's any issues, but it's been working fine.
Okay, shouldn't be
you.
any problem, and he has this, and he's not gonna use the clip.
Yep, I'm gonna stand right here.
He said he prefers that one, but
Okay, very good,
either one works fine.
where do you put the clipboard?
I'll put it back in the back.
Okay, and you wanna take it back?
Yeah, I can take it back.
This is his own laser point, I don't wanna steal his laser
Right.
point. Right?
Okay.
Do you want me to do anything once he finish?
No,
Just
I'll come down and take care of it.
Okay, we're good then.
Okay.
Thanks a
And I
lot.
have all the flyers and everything should be updated on
Okay.
the site. Let me know if you notice any mistakes.
Yeah, no, it was that, talk that was listed.
The April one, yeah, I fixed that.
Okay.
Yep.
Because someone sent me an email saying, oh, you're listed as cost of 2 talks.
I accidentally copied the one
And I was
before.
like, no, I'm not bringing so many people.
Yeah. Too much work.
Yeah. Hey, anyway,
Anyone sitting here? You may sit. You're going to get a cold zap. I walked through the town. He said by the time you reached here, temperature dropped from.
It's 58 degrees when I left.
Yeah.
So, when I got here, it was 28. But
You know, it was funny when I was driving back last night, it was like 40 in Roanoke. By the time I got to Blacksburg, which, you know, this is unusual, it was like 48. So you could tell that the winds were coming in, you know, from the north and breaking through. Normally, you know, we're a bit colder here, so it was interesting going up the mountain and getting warmer.
it went back down.
Yeah,
It went back down today.
it's coming through.
Depending on the wind.
Yeah.
And the tunnels are around in between the buildings.
Yeah, you can feel that, yeah.
When I was an undergraduate here, because I went to Cochran, and had most of my campuses, most of my classes in Auburn, all of us. So for Christmas, I said, "What would that play on this escalator? You say it was a big firm."
were you a BTA student?
Undergraduate, yes.
Yeah.
And my
Undergrad and PhD, both from here.
Biochem?
No.
biology and then plant microbiology.
Oh, very good, very good.
They went to William and Mary for his bachelor's.
William and Mary McQueen.
But we're right.
Much more advanced. Their labs down there were old.
You probably noticed that the campus has changed a lot.
Yes, indeed.
Yeah. I mean,
Yeah, I remember when this building was built. I
well,
was always fascinated with
in this campus you turn around and suddenly they build something.
Yeah, I know.
Yeah, and I knew the Fralins down in Rona, the real estate developers.
You lived into the honors house, right?
I was the first, in the first graduating class for the honors program.
Okay.
It's my undergraduate was here.
That's
and-
right.
and don't mention that one. But,
Yeah.
but I was in the first honors class, which was wonderful. Mr. Dudley,
Yeah.
Dr. Dudley.
He passed very
I
much.
know, yeah. He was my advisor. And Shirley and everybody and all.
Yeah.
It was so small that you could have.
Yeah.
I got to do my research in aerospace engineering, anthropology major.
Oh, wow.
They want to do something different. So it's a world space station. And it sounds sexy, but it really was just going in and put a little sensor on a little truss structure that mimics the structure itself. And then push a button on an old computer and wait 30 minutes. And do homework a little bit. then change it to another. That was a whole, all my.
Hey,
But it generated lots of cool data back then.
Yeah.
that time we used to do homework every time we have a gap, right? Now they check on the internet, they catch up with Facebook.
Yeah, exactly. Yeah, I had to read a book.
I remember those days when I run experiments and like 2 hours incubation, I was either running the second experiment or reading some paper.
Yeah.
Now you got social media and all
Yeah,
Trying
sorts
to
of.
yeah.
get
too
reading
busy catching
papers is the hardest thing you get them to do.
Hold on, I did
Why
some.
can I read the paper? I got Wikipedia.
Not only that, I've seen presentations from young students where they actually take
Yeah.
images from Wikipedia.
Yeah.
Also,
I'll fill that.
That's awesome. That's a
Best thing I've received.
this is for the questions at the end. No, this is, some students, they register, only a handful of them, they register to, for the seminar series as a course.
Okay.
So they actually get credit.
You get a taste for good.
Yeah. And I want to use this for attendance. I just
Oh, yeah,
don't
you say
want-
that.
We want to look into
I mean,
some
we
basic
ask you to send us
biopsy
some references and some papers, so we usually give it to them in advance so they read and know
Read
the
out.
research.
Okay.
So they're not caught by surprise when you come with one particular topic and then.
In mine, I don't get too deep into any particular topic here, so.
Well, you actually
No.
just did. Yeah,
No, it's fine, it's
Yeah.
just,
He was doing
I
some
like to
work on some
give them
of
some
the
heads up.
Hopefully we get back to see what kind of bacteria he's got. Yeah, we do see, I mean, it's amazing how the mixed genes and, or all the, you know, it's not just
So
but-
it looks like they have a different pharmacology.
Okay, so that's what it is.
Yeah, he's got some.
Yeah, yeah.
And he does anaerobic fermentation, so he can lower oxygen level.
No, it's
Yeah,
fine.
what level
I think that's really,
Yeah.
that's good.
he can show you that side.
You can microfile, sometimes soil can get atrophied.
Oh, yeah. Well, even just around the roots too, right, just because it's so high respiration.
And it's so much reducing, soil is so reduced, right?
Yeah.
The demonstration, medium with a surgery, soil clears immediately.
Yeah.
Boom. There's so much of reducing carbon.
Yeah,
Right?
yeah.
Yeah.
Yeah, he brings really unique perspective to cool biochemistry side.
You know, good universities, like this are the FralinLifeScience, virus.
Yeah.
They are looking slowly for mechanisms of more organismic and including ecological. They're starting to look for more mechanisms.
Yes, true, true. Of course, the mechanisms when ecologists is still different than the mechanisms.
I know. They are looking for molecular. Yeah, I was in a panel which was ecology, whole organism based, and two of us were there, we asking questions which are oddball, molecular mechanisms. I talked to the room, I said, are we doing anything wrong? No, psychological chemistry. He said, oh yeah. We just want to see slowly ecological work getting to the molecular.
We do have journals on molecular ecology, so.
So the idea is to collaboration, so
Yes.
that we improve the entire level in a project.
Yeah.
Yeah, to see that you can pick it up, right?
It's interesting, it's like my son, they always usually teach biology before you take chemistry. We even do that here. We tend to have students take biology before they take chemistry. It's like you should take chemistry first.
Well, in intro biology, you start off with chemistry.
Yeah.
Right?
Yeah.
At least that's what I would.
And I think if you're gonna understand biology better, you gotta know some basic chemistry.
Because chemistry leads to
Yeah.
DNA molecular interaction.
And also, if we be able to say, biology is chemistry and steroids, and elevate our field even to greater heights.
Well, other way to tell you that chemists will be out of job if biology did not come out.
Everybody,
Exactly.
Yeah.
every chemist I know is doing molecular
Has
biology.
to.
Yeah.
Has to, because chemistry is dumb.
Yeah, right.
All the basic chemistry.
That's what
Yeah.
I tell people. We discover something new in chemistry, in physics, something's changed in the universe.
then they do biomimetics, so that way
Yeah.
after biology has, why not? Millions years of experiment going on there.
Right.
Learn from there.
Just by chance, yeah.
Nature skills, right? So
Yeah.
Yeah, exactly. Yeah.
that's a sunny one. A lot of sun.
Yeah.
It's bright.
Yes, it looks nice.
It's a cool structure. I thought yours was really clever.
Yes. We made eye contact, we said hello, thank you.
Yeah, I thought that in front if you could record soybean.
We put it in, right? Big 7-foot tall.
I used to like draw, crop. I used to play inside, so I did. Yeah,
So
I'm from village, so I go and sit in the middle of the field, in between the rice or something like that. The only thing you get really bruised and bad.
Yeah, yeah.
But otherwise, it's a good place to hide.
So the thing you need to change is tobacco plant and you out there smoking a cigarette with a cowboy hat on.
Nice. And it's the law.
New Marlboro.
I'd be vaping. Heat not burn. That's what the Altria says now. Is
What's that?
heat not burn is the heat that Altria. So they've released it worldwide, but they sort of blocked its release in the U.S. But it's a little tobacco cartridge. And then you put it in this little device, it heats it up just enough to get the nicotine flavor, not burn out the other. So it's heat not burn. And they think that that's going to be their especially
With vaping looking so bad.
Although that's a different story.
Yeah.
But yeah, apparently, I know a buddy down works in Richmond, works with the he had one on the device, and he pulls it out, and it's like, played it in gold. It was really nice. Wow. And he said, this is the next thing. And they experimentally released it in 3 or 4 small countries,
Yeah.
sort of control it, and it immediately replaced normal cigarettes.
Wow.
Like, just
That's the growth area is Asia.
yeah.
There's more people smoking in Asia now than
Oh,
ever
just,
before.
yeah.
Ready? All right. Thank everybody for coming today. Special guest today, Dr. Scott Lohman. For those of you who are, how many of you are familiar with IALR? Just go ahead and raise your hand. Okay. IALR has been around for about 15, 20 years or so, and it's been under different management. So it's under really good management now. So we have it used to be run more, I think, by business people. Nothing wrong with that, right? But I think they needed a biological touch to get things moving. And so if you hear any negative things about ILR from other people, tell them, well, it's under new management now. So,
That's right.
and of course, Scott will tell you about how ILR works. I'm just going to tell you real quickly, he's a native from this region. He's actually from around the Danville region, which is where IALR is. And he did his master's degree at William and Mary. He then did a PhD here. And actually, that's in conjunction with IALR before he actually moved in and became the chief scientist and now eventually the director of the program. So he's going to give you an overview of the program. And one thing I want to note, he's already full with several meetings in the afternoon. He is going to have to get back tonight, so he won't be around for dinner. He'll be around for graduate students afterwards. But he does come back up, so you'll definitely be able to contact him at another time if you would like. He does come to Virginia Tech, at least once a month, I think. And so if you want to meet with him and you don't get a chance today, there's plenty of opportunities. So with that, Scott, take over.
Okay. All right. Thank you so much, Mark, for the kind introduction and inviting me up to speak at this at the seminar. So as Mark mentioned, the Institute's been around since about 2003. It was created as part of the tobacco settlement. Nationwide, the tobacco companies were forced to pay about $187 billion two different states. And Virginia got $2 billion. $1 billion went to the General Assembly. We're not really sure where that went. That was spent pretty quickly. Another $1 billion went to what's called the Tobacco Commission. And it's set up to focus on Southside and Southwest Virginia. And the Tobacco Commission then built 5 higher education centers. There's one in Roanoke called the Roanoke Higher Ed Center. There's one in the western part of the state. There's one in South Boston that focuses on IT and training. Microsoft's largest data center is just outside of South Boston, Virginia, and they focus really on supplying Microsoft with people trained in data management. And finally, there's one in Martinsville that is called New College. But we're the only one with research in our name. We're the Institute for Advanced Learning and Research, and that's what makes us different. We are what's considered a subdivision of the Commonwealth of Virginia, so we're not a state agency. At the state level, they don't see our budget. We just get a check from the state every year, which makes it really nice. We get about 78% of our funding from the state of Virginia. And so all the higher education centers were put there for economic transformation. As many may know, textiles, tobacco, furniture, all left, really less so for tobacco, but textiles and furniture left in the 1980s. Danville had one facility that employed about 15,000 people in textiles, and that just immediately evaporated. My father, I'm a first-generation college student from a little town called Alta Vista. My father worked at the Lane Company his entire life, and that also left and closed down. So I've not only believe in the mission of the Institute, but I've experienced part of that, the economic downturn that this was put there to replace. So what do? We do a lot of different things. We have advanced learning down there at the bottom in the green, and in it we have a mobile learning lab. It's about a $750,000 bus that has wet chemistry labs in it, and it goes around to different parts of the region and the state to show kids about STEM. We have the Governor's School and the Academy for Engineering and Technology, so we have about 250 high school students in our building every day for half a day. And then we have lots of summer camps and stuff to introduce kids to STEM, and our scientists work with those summer camps to encourage kids to explore STEM opportunities. On the workforce development side, we have a wonderful machining program that's about 80 years old, but we've taken it to another level. It's a model in the U.S. of training students to be not only great at machining, but also for management, and they run machines that are 5-axis CNC machines that are really built for components in the nuclear industry and in aircraft. So those students leave at 20 years old with a job making $75,000 a year, and oftentimes stay in the community. And finally, I don't want to forget, we have the conference services here over in the left. We have a wonderful conference facility, so we can seat and feed 500 people and give a presentation at the same time. We have an auditorium, all sorts of boardrooms for people to use, so a lot of activity takes place in the building. And as a scientist, we also have conferences there. So last year we had a bio-ag conference where we have folks from NSF and FFAR come down and visit. It really is a spot and a way up for us to get people together with common goals. And then finally, on applied research, I've got analytical chemistry, agricultural research, precision ag, and advanced materials listed currently, but we're always changing. Historically, we have been focused on agriculture. Agriculture is Virginia's #1 economic driver at $70 billion a year, and 335,000 jobs are involved in agriculture. So we, on the research side, focused on agricultural research, and on the workforce side, we focus on manufacturing. So those are Virginia's two #1 economic drivers. The advanced material, as an example of how we can help transform the economy through research, the advanced material group is actually, we had a company from France come in and rented all of our labs, and a lot of it was because of our connections with Virginia Tech and our facilities, and then hired our polymer chemist, and they're building their corporate headquarters for the U.S. in our park. So we're not doing a whole lot in advanced materials currently in the polymer science, primarily because industry we use it to leverage industry to locate in the area. But on the agricultural research side, Historically, we focused on biology and technology to improve agricultural production. I can get into it a little bit more, and I'll talk about each area in biology and technology, and also highlight some of our other capabilities and how we work with people, both industry and academia. And with that, I want to say that, we're not academia. We don't train students. So we get money from the state to do research, but we're not industry. We can't make products and sell them. So we play at the spot in between, and we found that to be a really nice spot to play. As Mark mentioned in the past, we did a lot of fundamental research that was terrific, but never really helped, and the folks in Southside never really understood what was going on. And then we sort of switched all the way over to trying to be industry and make things, and that didn't turn out well. So now we're back in between, and that's where I've taken the program and put the emphasis on it. So a little bit more background. All good agricultural presentations have to start off with this graph, right? To feed the world in 2050, we need to double agricultural production per acre. That's a huge challenge, and we have to do it on about the same amount of land. The green line here listed is the area harvested in cereal grains from 1961 to 2005. So we made wonderful gains from 1961 to 2005 on a per acre basis due to mechanization, breeding programs, but they were breeding programs that were focused on high inputs of fertilizer and selecting plants that could take that fertilizer and get larger and larger. As you can see there, but in the mid-90s, it sort of slowed down a bit. There was a peak at the end, probably GMO related. But nevertheless, those advances in mechanization and higher inputs of fertilizers and pesticides are not really a great option for our future. So we have to double agricultural production, but we don't have the tools really available that we had before. Now we have new tools like CRISPR and such that can make a difference. But at the same time, those, as I mentioned, the fertilizers that we're using, as we use more and more, they pollute the water. This is an example of a toxic algal bloom outside of Ohio, where the citizens of Cincinnati are even thinking about suing the farmers for putting it in there. So if you really want to change farming practices, start getting sued, and you'll try to use less phosphorus in this case. Nitrogen the same way. You see all the runoff here from all the way down to the Gulf of Mexico and causing those hypoxic dead zones. So we really need to figure out new ways to boost agricultural production. And one way we feel like that's been really ignored over the years is the biology of the plant and what's happening inside the plant specifically, and we call those endophytes. So we study bacteria that live inside of plants that can help plants grow. They can do lots of amazing things for plants. They produce hormones. One's called IAA. It's a growth hormone for plants. It's tremendous results that I'll show in a little bit. We have nitrogen fixation. They can help plants gather nutrients such as phosphate. They can help plants fight off diseases. They can help lower plant stress levels, plant stress hormone levels. So they can do lots of amazing things. And as we learn, we're learning more and more every day because the field was pretty much ignored until about 20 years ago. So what we did was we started the Plant Endophyte Research Center to sort of put our name on that field of study. The center itself, the institute labs were set up by a gentleman named Jerzy Novak. I was hoping he'd be here today. He's a pioneer in the study of beneficial bacterial endophytes. He was studying these bacteria that live inside of plants in the 1980s. And he'll tell you that at the time, most plant scientists could not understand or did not believe that there could be bacteria inside the plant doing good things for the plant. If the bacteria were inside the plant, it had to be taking something from the plant. We know that that's different now. And I want to highlight the people that do the work. Here we have Dr. Chuanqing Mei and Sajiva Amaradasa. They're plant biologists. Dr. May is our chief scientist. He has a wonderful background at the Noble Foundation, Michigan State, really molecular biology, assay development. Sajiva does a terrific job with all of our pathogens that we work with that I'll talk more about in the future. But we also have molecular biology, tissue culture, analytical chemistry. We have a drone pilot and plant phenotyping. We have a coding and robotics person on staff, as well as microbiology. So these people all contribute together with different skills and backgrounds towards this Plant Endophyte Research Center. And as I mentioned, the field of study is relatively new. As you can see here, back in 2000, there were hardly any publications based on plant endophytes. But what's interesting to me is it really mirrors the study of the human microbiome. As you can see back there, 1998 is really when the first publications started to pop up as far as the human microbiome. And I was just in Washington, D.C. the other day and met a lady named Pam Marone, who founded Marone BioVentures. She worked at Monsanto in the 80s. Monsanto was not interested in biologicals at the time. They were interested more in GMOs, which they, of course, were very successful with. But she pointed out something that I hadn't thought about before. 50% of medicines that we take have a natural origin. So they're somewhere or another came from trees or whatever. About 50% is what she stated, while only 4% of the chemicals and pesticides and stuff that we put on plants have natural origins. So we really have a tremendous opportunity to harness these bacteria to improve agricultural production. Now it's a huge challenge because we've bred our plants over the decades for high fertilizer inputs. Well, when you have high fertilizer inputs such as nitrogen, the plant doesn't need nitrogen. So bacteria in the plant sense that high nitrogen levels. So, fixing nitrogen is the most taxing reaction in Mother Nature. It takes 16 ATP to break apart one dinitrogen. So they don't do it unless they have to. So we've not only ignored that nitrogen fixation, we've selected almost against it for fertilizing. So we've got a lot to turn around and do, but you'll see in my next slides that we have opportunities here. So as I said early on, we did a lot of fundamental research. This is Jerzy Novak. If any of you haven't met him, he's a wonderful gentleman. He was my PhD advisor and a pioneer in the field. The bacteria that he really first found was Burkholderia phytofermens, strain PSJN. That's probably one of the most studied in the world. He first published about it in 1990. It was the first to have his genome sequenced. I know of at least 100 publications that's been focused on it itself. So when I first came, my background, I worked in the pharmaceutical industry for a number of years. I was interested in mammalian genetics, and I had the chance to get out of pharmaceuticals because I just didn't like the day-to-day corporate grind. And I co-founded an urban farm in Lynchburg, Virginia called Lynchburg Grows. And we set out to grow plants without fertilizers or pesticides. And let me tell you something, that's tough. It's not easy to do. So I became fascinated as a biologist with the microorganisms in the soil and how they could help. And then I met Jerzy, and he really enlightened me as far as bacteria that live inside the plants and colonize the plants to 100,000 colony forming units per gram of fresh weight. And when I first started working with this during my PhD studies, the results were just amazing in the lab in vitro. If you can see up there in the corner, the plants were twice as large after just 2 weeks, which is incredible. I'd never seen anything like that in agriculture. My background in genetics and breeding somewhat, just a little bit, told me that it took a long time to make a little difference in those type of programs. And to see something like this was just amazing, and to see how many were in there of just this one. So, to back up a little bit on the microbiology side, we can only culture between 1% and 5% of bacteria. So 95 to 99% of bacteria we've never studied, and we can't culture from these plants. So who knows all the different things that are in the plants and what they're doing. They found endophytes inside of plant nuclei controlling gene expression, for example. There was a recent published article in Nature where strawberries inoculated with a particular endophyte. The endophyte made its way through the plant into the flower, and then the bees that were pollinating, it went to the bees, and it made all of it immune to a fungus. So really, the different types of interactions that they have is incredible. They can control flowering time, we've seen. They can control taste. So just a simple inoculation made strawberries sweeter with one of our colleagues in Austria. Our fundamental research focused on gene expression. So we looked at microarray analysis when plants were inoculated with different bacteria and pathogens. That was most of my PhD research was on that type of stuff. And then we'd go back to the lab and knock out genes in switchgrass, and we'd overexpress genes and see how they responded. We've looked at horizontal gene transfer of the nitrogen fixing enzyme. We've looked at DNA methylation upon colonization. A lot of fundamental, really interesting stuff. But about 3 years ago, when I took over as chief scientist, we sort of wanted to take a little bit new direction because we have funding from the state, and there's not a lot of funding at the federal level for beneficial bacteria. You know, most of the work in plant microbe interactions and that type of program is focused on pathogens, which is rightly so. And industry itself is not necessarily funding a lot because these are naturally occurring organisms. You know, industry wants to take a chemical, get a patent on it, and you know, make money for 15 years, basically, that's their model. They have to pay the bills, and there's nothing wrong with that. So there's not a lot of funding for this type of research. So we thought, well, why don't we take our state funding and invest in something that's got value that we can then go back and work with industry and academia, and so this is very much applied research, but at least the fundamental questions. I don't want to go through all this, but we had a very pragmatic approach to isolating these endophytes, to address ownership. We utilize our analytical chemistry component, all of our different skill sets in molecular biology and in assay development and such. And today we have about 2,000 bacteria that we fully characterized, and they're based on 12 different plant growth promotive properties, way more than you see listed here. I've listed ACC deaminase, which lowers the level of ethylene in plants, which is a stress hormone, makes the plants larger. IEA production, nitrogen fixation, and phosphate solubilization. We looked at siderophore secretion. I can't even think of all the ones off the top of my head, but we also look at antifungal and antibacterial production, and we looked at specifically at what pathogens those isolates can work against. So we have a plant pathologist on staff now that I'll talk about more in the future, and he's collected 30 plant pathogens that we screened all these 2,000 bacteria against. So we have a wonderful collection of all this data of what these different bacteria do, and many do lots of different things. So just as an example, this is auxin production or IEA production. We were at 1950 here. So we take these top performers, the 50 or 100 top performers, and then we take those to the lab and we do in vitro growth assays. Now you can see here on the left axis is 100%, 200%, that's percent increase in growth in biomass. As you can see, one of them's up to almost 900%, 9 times larger. It's really incredible to see. There's PAKM down at the bottom. We've published this data. Just by simply inoculating with a single bacterium that produces high levels of the plant hormone auxin can increase the plant growth dramatically in vitro. So these are some really interesting numbers just as an example. For phosphate solubilization, we took those top performers and then we grow them in the presence of insoluble phosphate. So the bacteria has to make the phosphate available. And we also see some pretty significant increases in growth in phosphate limited soil. And then if this little dotted line here, which I may be able to highlight, but maybe not, the little dotted line here, the other side is in greenhouse. So that's greenhouse studies. So my point there is that oftentimes what we see in vitro is diminished in the greenhouse when we go to soil, potting soil. You know, this is not sterile, and it's diminished further in the field. But nevertheless, the results are stark. It's really interesting to see these plants. If you can see up there on the top, it looks like literally they're on steroids. So that's another example. Here's a couple examples of ACC deaminase. Again, that lowers stress hormone levels in plants. IEA, and then when we put them together, and there's not necessarily a benefit when we co-culture two different ones. And we found that out. You know, we've looked at, why don't we make consortia? Because the idea is you start adding the bacteria together, you expect the results to increase. But a lot of times it doesn't work that way. In fact, the results will be worse. I don't know whether it's competing or whatever. We have a lot of stuff to learn. And then I mentioned plant pathogens. So this is just some of the plant pathogens. This is where we were last year. Sajiva goes out and collects the pathogens from farmers. We work with Virginia Tech's plant pathology lab. And then he screens them against bacteria. And the interesting thing to point out is we have over 2,000 bacteria now. But look at it. Some of them only 5 bacteria work against them. So it's not a lot that that these things, they're very specific with their biocontrol properties. And then he does both greenhouse and in vitro work where he actually inoculates the plants and sees improvements in plant growth.
Sorry, I missed something there. So what you're saying is that you have bacteria that can actually prevent the pathogen from actually infecting those plants?
We're not. I wouldn't want to speculate that. The plants survive better when they're inoculated. Yeah, their mechanisms, and see that's a fundamental question. Yeah, and this is what we look for initially. The screens are just the beneficial bacteria and the pathogen, and then you can see the zones of inhibition. So then we take those to the greenhouse to actually do in-plant studies. As a great example of this, Dr. Jaish Samatini there is part of Cooperative Extension, and he came to us and knowing that strawberry farmers were losing the use of methyl bromide. Methyl bromide is a cancer-causing agent that's a soil fumigant that kills microorganisms, pathogens in the soil. Probably kills good bacteria too, of course, but it kills pathogens in the soil before they plant strawberries. Because they can no longer use that, their yields go down 20% almost immediately. And then when you have a lot of strawberry farms are agrotourism farms, in other words, they have their crops in the same spot every year, they can't rotate. They have to have it there where the public can get to it. We think the load may increase over time. So he came to us with that problem, and we worked with NC State and Virginia Tech, and we got the 3 most common strawberry pathogens as isolates, and we screened their bacteria against them. Well, Sajiva, who's in charge of that, we only found 3 that worked against all 3 bacteria. So we took those to the greenhouse, as you can see the second line here, where we had a lot of increase in fresh weight and survival rate. We used, at the same time I should say, we went to the East Coast largest supplier of strawberry runners. So we don't want to just do this in the lab, we want to do it with industry. So we went to Aaron's Creek Nursery, which is in Virginia, and they supply strawberry runners to growers from Michigan, down the eastern seaboard, and into Texas. So we got true strawberry runners from them as strawberry growers were plant, and that's Elon, Sweet Charlie, and Chandler. And you can see the increases in the greenhouse. And then we went to VDAX then and got funding to do field trials. So we did 3 field trials across the state, and we saw about a 15% increase in marketable yield, which is the most important thing. We didn't look at biomass. We wanted to know how many good strawberries they were getting off those plants. So in 1 year, that's what they were losing with methyl bromide. We found our own money now, and we've repeated those studies. And then this is something that we can take to a company that would be interested in marketing it, because again, we can't make a product, but we can develop products. A second great example is Dr. Brent Nielsen from BYU contacted us, and he, of course, BYU's at Salt Lake City, and I flew out there, and it indeed has high salty soil. If you can imagine, it is salt. And they only have a couple of different plant varieties that will grow in it. And he isolated endophytes from those and then started testing, and we were able to show through our own data down at the bottom that A07-1, that's rice. So it increased salt tolerance in rice. And then we just published in Frontiers in Microbiology about that work. So he came to us with that idea. We did some preliminary data, and now we're applying for 2 separate grants to fund this work further. So it really is a partnership, a mutually beneficial partnership. I've got a nice blank slide here because we do tons of work with industry, but we have NDAs, non-disclosure agreements with each. So I don't really want to put it on, advertise it too much. But we work with Indigo Ag, which is a disruptor, if anybody's heard of Indigo. They focus on endophytes, and they license 50 of our best strains right away. And if any of those become products, we get money from that. We work with Ag Biome, with Novozymes, with large companies like that. But what we particularly like to...
I have a question about that. How can you license something that is a natural organism?
It has value, and it's more like trade secret versus patentable.
Oh, I see.
Yeah. It does so well that they'll take it and run with it. And Pam Marone did it with...
It's not a patent.
It's not a patent. No, you can't patent a naturally occurring organism. Now, what happens is they'll put them into a product. They'll have a process, and that process they'll patent with the bug. So that's sort of how they get around it. But We work with like small companies. There's a couple startups out of Charlottesville that we work with. Of course, UVA, as good of a university as it is, doesn't have any really plant science facilities there to speak of. So we have some biochemists from UVA that have created what's called an Agro Sphere. And essentially when bacteria divide, sometimes they divide unevenly, and they'll just create a sort of a sphere. They figure out how to program bacteria to do that, and then they can place things in that sphere. And what it becomes is an extended release delivery system. They can put chemicals in it, they can put biologicals in it, and they spray it on the plant, and it actually sticks to the plant better than just a chemical alone. So we do most of their research for them and contract research. And I'll talk about the different ways we work with the industry at the end. Most exciting, I think, is we've had 2 companies that we've finally got all the legal work done for the MOU, but we've become their discovery arm. So there are a lot of new startup companies out there now that they have technology for delivery. For instance, one, there are 2 types of bacteria, they're gram-positive and gram-negative. Gram-positive bacteria form endospores a lot of times, bacillus. So terrific shelf life, you can use them anytime. But gram-negative bacteria are much larger, much more diverse in their genomes, but they don't survive well, so they have terrible shelf life. Well, this particular company that we work with has a way to preserve them in a product, but they don't have any bacteria. So we've become their discovery arm. We will send them their bacteria, we'll send them our bacteria, they'll put them in their product and send them back to us. We'll test them. If any one works, it can go to market. We've got a separate project that's replacing, if any of you have heard of Serenade, it's one of the world's most used biological fungicides. It was discovered first by Pam Marone in the late 1990s. She sold it to Bayer for $600 million and got a spot on Bayer's board and got to keep her company. That is really towards the end of its life now. There's several people making it, and it's no patent protection. Anyway, a company came to us and said, "Can you find a replacement for it?" And so we screened our bacteria, and for broad spectrum plant pathogen inhibition, and we have 11 that may very well fit. So we're going to send them to those folks. They're going to test them in their own greenhouse. If it becomes a product, we get a royalty stream. So those are just a couple examples of projects that we have with industry that are really unique, and there's a lot more. So I think we're operating under about 15 NDAs currently. And what we really want to be is a spot where, as academia, if you have some product that you think that industry would be interested in, we can be the conduit to that because we have connections with all these different industrial partners that can take biologicals to market. So we really want to be sort of like an ecosystem as well, because we don't care if someone has something great and it can be great for a company. So if a professor has something that he's discovered that's awesome, was having trouble getting in contact with companies, we can make that connection. We don't need anything from it. We don't worry about paychecks or payroll or grants or anything like that because of the way the state set us up. So we really want to be sort of an ecosystem. So I want to switch gears here real fast. And how am I doing on time? Pretty good. And talk just a little bit about some of our technology platforms. So we work with mechanical engineering at Virginia Tech and develop these smart tables. And they are 3-axis CNC gantry robotic tables that are accurate to within a thousandth of an inch. So essentially, well, let me go through this first. I'll have a video here next of it running. But it takes images of plants. And because it's accurate to within a thousandth of an inch, it's like having a fixed camera above every plant on the table. You can then take and introduce treatments to the plant or take away treatments or do something to the plant, and you can watch dynamically how it responds. Now, when it really becomes powerful is when you combine that with computer vision and robotics and coding and get data from it. So images themselves are multidimensional. There's a temporal decoupling of sampling time and analysis. So the experiments we did 5 years ago, we can go back tomorrow with new coding and new whatever to look at different things. So it really is a permanent experiment that you can look at different ways. And it's really excellent at measuring phenotypic responses over time, and I'll show you a few examples of those. The tables themselves, let's see. As I said, they're accurate to within a thousandth of an inch. So essentially, we can time lapse every plant on the table growing. and then watch how it responds. And it'll run 24 hours a day, 7 days a week. So it doesn't stop collecting data. And it's really amazing how accurate the thing is and precise. We can also screen our bacteria against it, so we set up floating tray systems. So that's 1,300 plants on that table that we can introduce treatments to and watch and analyze and do. And this is the type of data that's generated. This is very early data that we had. This is back in probably 2017. But that's the type of the data we can see on this first chart. We can see the blue line is a control treatment. And then the other 3 lines are different beneficial bacteria that we have that perform well in the lab, in vitro. So, we inoculate it, and at the end of the study, we weigh it, and they do well. But this you can see almost after 10 days, we can see if the treatment's working. So at 20 days, we're good. We're done with it. We can see the type of increase in plant growth. And then the way we do that is through image analysis and looking at we count the green pixels, essentially. But what's amazing about it is the ability to time lapse. So this is one of our first studies we did with Dr. David Reed in 2015, and these are tobacco seeds germinating. And we only capture data once a day on these. No, wait a minute, 3 times a day. And what's interesting to me is this was I think it was a 2-week study. But anyway, that robotic arm moved. We calculated up 250,000 times in 2 weeks. And it only knows where it is based on where it was last. So it's controlled by what's called stepper motors. So they do steps in between. It's so accurate that even these are floating trays, that it's almost a perfect time lapse. The only thing you see is a little bit, sometimes the water went down a little bit, that the tray went a little bit away from you. But we was like, wow, that's really amazing that thing is that accurate that you can time lapse everything. And then of course we can generate the data from it for germination. But here are a couple examples of just how precise these things are. So this is a Passionflower plant. It's a climber. And that was one plant out of 20 that we were watching with the system. And what was interesting to me is that one long tendril there stopped, and then the young tendril started. So how does the plant know to stop one and start another? So we set up another experiment. And this is my favorite video of all time, by the way. Politicians have seen this. I've shown this to everyone. This is also a passionflower plant attempting to climb a pole. And I believe that this plant makes a decision. So watch. So it's attempting to climb it in a clockwise fashion, but you can see its little tendril is missing the top. You can almost see the plant get frustrated if you watch it enough and watch what it does. It goes around the other way and successfully grabs it. So when you can start seeing, and then the plant, it looks like it's happy then. It's like, I made a good choice. But you can see sort of the detail that you can see in plants. So we can see leaf tip curling. We can see curling along the edges. We can see color changes over time. And we can measure all those because we have a computer vision robotics person. So Daniel took that same image and then figured out a way to sort of estimate movement and put numbers to it. And then that's what that graph looks like. So it's moving, moving, moving. And then when it makes that abrupt move, it goes up.
Did you put it to YouTube?
That's on YouTube, yeah. Yeah. If you look it up, share it, hashtag us. I'll probably say that wrong, so don't laugh at me. So then we want to see, well, can you relate it to plant health? Can you relate plant movement to plant health? Bingyu Zhao, who couldn't make it today, is a wonderful partner of ours, and he knew of a person that were able to show that when plants were introduced to pathogens, their motion changed. I don't think they were really putting numbers to that, but nevertheless. So here's a good example. Let's see if this works right. So these are tomato plants recovering. They sort of get going, then we add fertilizer. And you see, of course, how much the plants start moving. And then that's what the graph looks like there on the right. And then we took peppers and a population of peppers and introduced different treatments to it. And then this is what the data looks like. So the blue is control on this side,
And this is plant growth. This is how fast they're growing. The yellow line, the yellow dotted line here on the bottom, it doesn't work. This yellow line here is heat. And then both of the other two lines are nitrogen, 600 milligrams of ammonium sulfate per liter. So obviously those grew faster. And both of the heats trailed off at the end because the plants dried out, so drought. So we got that on the same chart. But look what the data looks like. It's pretty precise. And then, but look at plant movement. It's completely different. So the controls down at the bottom sort of moving the slowest. Both heat treatments move the most, and heat only tended to have the highest movement. And then you can see over there the data that's collected.
Where's your heat treatment? I mean, can you give us an idea of the magnitude?
A mat, and I don't have the temperature, but it was like 98 degrees. It was pretty warm, but it's a mat that you germinate seeds on.
I see.
Yeah, and these are just proof of concept. What happens when we do this type of thing? But heat only moved it up a bunch, and then here, these are pathogens. So we took 3 plant pathogens, Fusarium, Pythium, and Agrobacterium, and introduced it to bell pepper. And the top line is the control, and the bottom line, you could see them, they just didn't respond as well. So this is just really sort of basic preliminary data. We are about 3/4 way through a paper that we're going to publish, and we think it'll be the first of its type. And we looked at beneficial bacteria, pathogenic bacteria, and a tomato plant that was susceptible to that pathogen, and then how they both grow and motion behind it. It's really fascinating. So we're really excited about that. We just can't quite get it out the lab fast enough and publish, but we're working on it. Everybody here knows about how that goes.
Why is that the plant, maybe you said and I missed it, why is that the plant movement is, sorry, I'm not a plant person, why is that the plant movement is so important in your readouts?
Well, most people think that, typically people don't think plant move much. You would expect them to just grow out like this. So just being able to see that they're shaking as they're growing, but there's no explanation that I've seen published as to why plants shake as they grow. But you can imagine with the Passionflower plant, it has to move because it's a climbing plant. So it has to reach. And if you think about it, it would need to be able to make decisions because you just can't sort of randomly reach for things all the time. It needs to be informed of something like that. But I've never seen anything published, and I think we'll have one of the first publications on plant motion in relation to plant health. But the bottom line is it's a brand new field of study.
So
And
more movement is a healthy plant.
yes, faster growing, essentially.
And what kind of time scale? I'm sure it's a time lapse. So what kind of time scale those movies are?
Different for different ones. This is, let's see, that's over days there. The Passionflower was just 1 day. That was a 24-hour period. So they move differently. We don't have a standard time that we look at it, but it's just fascinating. So what we think is that plant motion stops. So when we introduce a pathogen, plant motion stops immediately, right here. But plant growth doesn't. It takes a couple days to notice that. So it's also an early detection of plant health. So imagine in the future, like indoor ag, robots or computer vision could be watching the plants and see how healthy they are, but basically on how fast they're shaking. Because they can see that, we can obviously.
Is there any way if you could connect that to energy production, or be useful how much energy?
I don't know.
Yeah.
Yep, something to consider. We hope that once we publish it, we'll have opportunities to work with others. Another program that we have that I'll just briefly touch on, we have about $100,000 worth of drones, and we've flown 400 flights over fields in Southern Virginia. We can do NDVI, normalized difference vegetation index to see plant health. We can do 3-dimensional modeling for yield, and these are the different farmers we'll work with. So they have become a really tremendous source of both ideas and as a resource for field trials as the farmers themselves. So now we've sort of moved away from just introducing the technology to farmers, and now we use it more for field trials ourselves with our bacteria. Another thing I want to point out is that we have really a wonderful analytical chemistry lab. We have 2 UPLCs, ultra performance liquid chromatographs. They're both matched up with MS detectors. One's a triple quad, which is tremendous at quantification. The other's a QTOF. It's and I'm not a chemist. It's a quadruple time of flight, and it's really terrific at identifying compounds and proteomics. We have a GCMS from Algent. We just purchased a new ICPMS, which is inductively coupled plasma spectrometry. Anyway, so we have wonderful equipment, and what we find that's interesting is that even large companies like Novozymes, they typically don't have this type of chemistry lab, because these are obviously, each of them's a $500,000 piece of equipment with a $50,000 a year price tag just to keep them running. And so we do a lot of chemistry analysis for different companies. And that's really where we found a really sweet spot to help companies out in the next, moving things forward. So like Agro Spheres, we can quantify what stays on the plant, what doesn't, some residual assays for different types of pesticides that we can do. So that's just another resource we have, but it really matches nicely with the other things that we're doing. And then finally, this is a brand new initiative to us. Dr. Michael Evans there, who's the director of the School of Plant and Environmental Science at Virginia Tech, he came down and had some money, and we are converting this greenhouse here into an innovation center. We went to the Tobacco Commission and matched all the money he had and put in our own money. It's about $1 million project over 3 years. And we'll be looking at controlled environment ag, so things grown in the head house versus the 2 greenhouses. And there's some examples of it there. What's really needed to us, of course, we're there for economic transformation. Almost the time we submitted this grant to the Tobacco Commission, a company called AeroFarms announced that they're building the world's largest indoor ag facility 3 miles from our campus. And I just met with the CSO yesterday. They're interested in working with us to develop grant proposals and other things to, like the Future for Foundation of Agricultural Research. It's really nice that they fund the program. So having them working with us to increase production would be wonderful. Their facility, I believe, is about 100,000 square feet. It's a brand new facility out of the ground. They grow 12 levels high, so it's 12 levels on top of each other. They grow lettuces, and they'll grow 2.1 million pounds of lettuce out of that facility in 1 year. They harvest year-round. There's no pests, so there's no pesticides. The lights stay on 24 hours a day. They can harvest the lettuce every 18 days. So if you can see, that's a manufacturing facility. But they're located right there next to us. This center, we wanted it set up to work with industry to test different things. We're going to be integrating our biologicals into it. You know, as I mentioned earlier, in the lab, our biologicals are terrific. In the field, they're not as, you know, the results aren't anywhere near as stark. But we think that the hydroponics in a controlled environment may be more like the lab than the field. So we're really interested in seeing those type of results. And so to recap, at the Institute, our research program is, our research sort of investment strategy is to invest in initiatives. So like the endophytes that aren't well funded, that we can be the leaders in, the computer vision robotics, the equipment like the triple quad, everything that we're putting into the innovation center, the drones, and then the skills. So we get people with different skills that can contribute as a team, and of course collaboration spurs innovation. With industry, we can do contract research, so our contract research is cheaper than they can do the research itself. We don't charge machine time, we just charge for supplies, and then man hours at a very affordable rate. So we do lots of contract research. We have licensing agreements like with Indigo Ag and others. We can co-develop IP like the 2 of the projects that I talked about. We rent lab equipment, we rent benchtop space. We can do workforce training, so we can help if a company needs X type of skills, we can bring people through. We have low-cost testing that we can do for the folks. In academia, we can generate preliminary data for mutually beneficial project with some of our advanced equipment. Grant co-development, matching funds like from the Tobacco Commission that we did with Michael Evans. He had some funds, we were able to double those. And then commercialization. We have a number of different innovative projects under development. One with Biswarp. We have a fermentation facility that we like to put in. It's just a natural fit to our biological program. But at the same time, what's interesting is when he came to us with that, he wants to focus on industry and attracting industry to the region. So as you know, biological manufacturing is something that's going to be more and more common in the future. And this is a way we can sort of position ourselves to be leaders in that and be part of that ecosystem. And then with Mark Williams, we're developing some nitrogen fixing stuff that's really interesting. So plants gathering nitrogen from the atmosphere, not lagoons, but grasses and other things. So with that, I will conclude my presentation and see if you have any questions. Go ahead.
That's really exciting. Thank you for
Uh-huh.
sharing all that. According to your endophytes, do you do any like fungal endophytes or just bacterial?
Just bacterial. We'd love to get into the fungal part because I think there's some synergies. So we've done some early work with AMF, fungal microorganisms, and colonization. And we've been able to show that beneficial bacteria actually can aid that root colonization with AMF, which is kind of cool. But there's lots of opportunities. The problem is it gets so complicated so quickly. Just 3 bacteria together, you think, well, by the time you do the experiment and all these different results and stuff, but that's somewhere we would like to move in the future for sure.
Yeah, I have 2 quick questions. One is, it seems that this is working okay for the scale where the plants are small, and, right? I mean, it's like you still haven't really moved this towards wheat or...
Yeah, we did switchgrass early on, so we published in field trials in switchgrass, and it was a 15% increase in yield. A lot of it's the plants are more healthy when they go to the field type of thing.
And the other one, I had the impression, I might be wrong, but I had the impression that I heard about the Lynchburg growth.
Okay.
Either radio or TV or
Uh-huh.
something.
Yep.
That it was kind of a very interesting initiative because
Yep.
of all this, pesticide-free stuff. I don't think-
Yeah, so that's my baby.
Did you?
So I love to hear good stuff about it.
Yeah, I had the impression that I heard something that would it be possible that to increase the productivity or something you were planning to put some robots and grow the plants in some kind of multi-layers platform?
So that's what Air Farms does. We tried it at Lynchburg Grows. We tried aquaponics, where you grow fish and plants together. That was a nightmare. I don't even want to. That keeps me up at night thinking different ways we killed fish. So I stay away from that. We did some little bit of vertical farming at Lynchburg Grows. Yeah, Lynchburg Grows is we have 70,000 square feet of greenhouse space in Lynchburg. Some of the greenhouses are 100 years old. It was a flower production facility. They grew long stem roses for 50 years at least. And they were 23% of Virginia's flower industry as far as dollar volume. They would harvest 10,000 long stem roses every day at a wholesale cost of $1 per stem. So if you can imagine, that's when agriculture in the U.S. was really rolling.
Next week is going to be really good.
Exactly. So, but NAFTA, the North American Free Trade Act, really decimated that because it opened up flower growing to Colombia and Ecuador, where they can go on the side of a mountain. We decided in the U.S. that our sweethearts needed flowers on Valentine's Day. Well, that's February. So that means you have to grow the roses all winter. And so there was always a lot of input of heat and pesticides and such. Well, where down in Colombia and Ecuador, it's 70 degrees all year round. Labor is almost no cost. So the price of roses went from $1 a stem to 25 cents a stem overnight. And there used to be 400 rose growers in the U.S., and now there may be 10 at the most. And Mr. Schinkel, who graduated from Cornell and his dad ran the farm, then he ran it, testified before Congress on behalf of the flower industry about the decimation. But their loss was their gain, because once they went out of business, they sold us the whole farm at a reduced cost. We have a farmhouse, and what we do is we have job training programs for people with disabilities. So we have about 20 people with disabilities that work there and harvest for us. We have 150 people in our CSA, Consumer Supported Agriculture Program, and that's how we keep the farm running. So we've been in operation for 18 years as a private nonprofit and have never missed a paycheck. So I'm very proud of that. No, not currently. We work a little bit with Angela Sasesic for the Austrian Institute of Technology, but specifically with biologicals, other countries are more restricted than we are. So we can't. They don't want any biologicals coming from any other country. So it really does make it challenging in that aspect. But we're always interested in working with others. That's for sure. We can do it for sure.
So you are catering to the industry, but now you have drone. Can you give service to the farmers?
Yes. So that's farmers are industry. I love those all together. And so we still, if someone calls us and has a disease problem or disease question, or if something's going on weird in the field, fields are interesting. Fields, one part of the field can be 3 times as productive as others. That's a lot of great data. So all those drone flights we did, we worked with the farmers to say, hey, do you have any interesting things going on? We want to fly those multiple times. We have a lot of times 5 different series of flights over those challenge parts. So we have all this wonderful data. We're doing a lot with hemp, industrial hemp. So we've tested for 300 growers this past season. So we're trying to facilitate that as well. And I mentioned the conference service facility that we have. We have a conference in 2 weeks. That's the 3rd industrial hemp conference we have that 450 people will attend. So we're really trying to facilitate that industry as well.
So you're getting data from them that what they're doing to the field so that you can integrate with
Yeah. But really we found it more just as a tool in our toolbox that as we need to go out for field trials in the future, we can use those drones. There are multispectral cameras and thermal cameras on those.
No more questions? Okay, with that, let's give Dr. Lohman another hand. Certainly, like I said, if you want to talk to him afterwards, great. But and he's gonna go eat. Are there any graduate students who are interested, signed up to go out for lunch?
Okay, teacher. Good to see you. Okay, yeah.
Please get, I'll remind you.
Okay.
Get the flyers.
Yeah, right, yeah.
Because now time to get active.
That's right, yeah. Thanks.
Yeah, I've been meaning actually to send you off, so we have a couple of new isolates that are like, there's nothing in the database about them, they've got some Pathanophaga.
Okay.
That if you guys could, do some macropodic, looking at it, see isolates, just run some of your assays on
Yep.
them.
Oh, yeah. It plugs in really easily.
Yeah.
It's very easy to do.
Dr. Lohman,
Hi there.
I'm Kelsey Reed. I'm a grad student here.
Uh-huh.
I was just curious, do you guys have grad students come out to ever visit the
Yes.
institute?
Uh-huh.
Okay. How would that get set up?
You can just
Like, can
email
I
me.
email you?
Yeah.
Okay.
Mhm.
We have a space for lunch if you want to have lunch with me.
I have class at 1:30. You're also Wood.
Yeah. Please do. Just email me.
Okay,
Yeah.
perfect. Thank you so
Uh-huh.
much.
Hey.
Very nice presentation
Okay.
there. And I asked you about that because I'm a professor in Brazil.
Okay.
In a
Oh,
university
wonderful.
there, And we
Yeah.
do research on agricultural a lot. It's a
You-
very
you're
strong
way ahead of
university in
us.
Brazil.
Wonderful.
Okay, so the idea is to, I understood that maybe you can help on providing fundings or connecting to industry
Mhm.
needs, and so I took a picture of your contacts here,
Okay.
so I can maybe I'm gonna send your contact to someone in Brazil so they can
Okay.
make the guys from the agriculture.
Yeah.
department there.
We can house graduate students if they're interested in that. Any way we can work together. I've always admired the work that you guys did with sugarcane and nitrogen
I
fixation.
actually work with sugarcane.
That's wonderful. Yeah, that was early on with my studies. I was interested in that, and then with soybean as well. I think
Soybean
selecting
with-
was it Johanna
That's why I remember.
Dolfsberger? I can't
And it was
pronounce her name, but she was really a pioneer in the 60s and 70s with selecting soybeans for nitrogen fixation.
Who was that?
Johanna something.
Johanna.
She's really a pioneer in the field of microbes and plants,
Okay.
to me at least.
Like
And she did
in the
res-
university where I work, we are very strong in microbiology, in agricultural,
Mhm.
like field agricultural, and horticultural,
Mhm.
like tomatoes and everything.
Sure.
Like these soybeans is very important. Like
Yeah.
many of the cultivars that were developed in our plant in Brazil were developed from a researcher, a breeder from our
Oh, okay,
university.
wow. Okay, nice.
His name is Tuneo Sediama.
Okay.
Tuneo Japanese. Tuneo Sediama. And now we are starting working with images and trying to make have a facility to take pictures like this,
Yep, mhm.
images.
Yeah, wonderful.
Working with that, drones and everything.
Very cool.
And I'm
Yeah,
trying to apply that with sugarcane breeding. So we need collaborators
Yep.
in this part.
And there's multinational opportunities for funding too. So yeah, please contact me.
Like-
Because
like-
I always reference Brazil as being a leader in
Like
microbiology and plants
Yeah.
because we've ignored it here in the U.S. for decades.
Like yesterday, I had a meeting with the people there through Skype,
Mm-hmm.
and we are about to enter in a call from our government on artificial intelligence applied in many fields. One
Okay.
of them is agriculture.
Nice.
So we need, like we have a FAPESP from São Paulo, Gota knows FAPESP, and another funding agencies. And it's like we need to find some industry
Mhm.
that will put the same money as FAPESP puts.
Yep.
Like if they put 1 million, we have to find
Yeah.
industries that will put the same.
Yep.
So now we are and we have until May to find some industries or some companies that would like to provide some
Matching
funding,
funds.
to
Yeah.
match the funding.
Mhm. Yep.
So
Yeah, wonderful.
that's the point. So I'm gonna send you an email,
Okay.
and then maybe you can help us with that, and
Yeah.
depending on the interest, we can make some
Exactly,
kind
yeah.
of research.
Mhm.
That's maybe some research that's going on that's may interest you.
Oh,
or,
sure. You
know.
like I said, you guys
be
are
much better.
far ahead of the U.S.
Yeah.
in microbiology component.
Uh-huh.
Anytime when I used to present, I would always brag about if you want to see how to do it, go to Brazil. Look at what they did with soybean and
Mm-hmm.
with sugarcane. Because there's still a lot of folks in the U.S. that don't think you can fix nitrogen without a nodule. There's
Mhm.
still a lot of plant scientists that believe that.
Okay.
And I think sugarcane gets a good amount of its nitrogen from the atmosphere, if I'm not mistaken.
Yes.
Yeah.
Yes.
Yeah, and it's not nodule.
Yes. Sure.
Yeah.
All right.
Okay. Well, thanks. But
Very
please
good.
email me, and I look forward to
Okay.
further conversations.
All right.
Yeah.
Hi, my name is Kota Morota.
Kota?
Yes, I'm a faculty member in Animal and Poultry Sciences.
Okay.
And also hosting him for
Okay.
this Virgin Tech. So we're interested in doing application of computer vision system.
Okay.
So livestock.
Ooh.
We do plants, but also animal side.
Okay.
Yes, particularly for estimate body weights,
Uh-huh.
image 3D image data,
Okay.
and also monitor animal behaviors
Mm-hmm.
for animal welfare purposes.
Okay. So if you need like preliminary data
Mm-hmm.
on a farm that you have, if you want a drone
We
pilot
are
to come up.
currently collecting in the Virgin Tech.
Okay.
And you mentioned that there's someone in your group who
Mhm.
does computer vision system.
Yes. Yeah. mhm,
Okay.
Yeah.
Yeah, I was wondering if you can put me in contact with.
Yeah. If you don't mind emailing me,
Sure. Okay.
just email me.
Mhm. I will email you.
And I will
Okay.
get you in touch with Daniel. He's a computer science guy, so that he's
Okay.
not just a plant person,
Mhm.
and he likes challenges,
Okay.
and then can do what he can. That's for sure.
Okay.
Yeah, if there's opportunity for code grant development, that'd be
Mhm.
wonderful. But just preliminary data is also awesome.
Okay.
Yeah.
And then at the IALR, do you mainly focus on plant side, or are you open to other?
We're open to other
spaces?
things. Yeah, we can go
Okay.
where we want to, but
Okay.
we just focus on plants because that's their
Okay.
expertise.
Mhm.
But partnering with someone outside of plants is just fine.
I mean, we ordered it.
Yeah.
I mean, we're gonna.
Okay.
Thank you.
we're gonna-
Yeah, enjoy your talk.
Thank you.
Yes.
Okay, if you wanna go,
lunch here?
we already have it, we have it all there and everything. I mean,
Oh, good.
Instead of chatting here, okay, great, we ordered it, otherwise, let's see if we want to take it. Scott, you can leave everything there.
Okay, wonderful.